In this video, I'm going to go ahead and talk about where the first cells came from. Previously, we said, according to the cell theory, that all cells come from pre-existing cells, but that really doesn't answer the question. What came first, the chicken and the egg? Where did they come from? Well, let's first of all talk about what the Earth looked like a long, long time ago. So, hundreds of millions of years ago, before there was life or cells, what did the Earth look like? It's what we call a reducing environment, very, very different than what we have today. We have an atmosphere that pretty much consists of nitrogen and oxygen. Not so long time ago. Early Earth was thought to be very hot and wet, and the hot temperatures vaporized liquids. So we had oceans, but a lot of steam in the air because it was very hot. Lots of volcanoes. The atmosphere was not stable, so there was a lot of lightning. It was ionized. And the gases, in particular, were very, very different. So instead of having nitrogen and oxygen, we had lots of gases that we call reducing gases, like methane, hydrogen, carbon monoxide, carbon dioxide also, but very little oxygen in there. How did the first cells then get going in such an environment when they really didn't have what they needed to sustain life? Okay, let's go ahead and look at the 4 conditions that they would need. So in order for living things, primitive cells, to get going here, we have to satisfy 4 different things. First of all, you need to make organic molecules from non-living inorganic matter. That's going to be a real trick. The second thing is we need to go ahead and assemble those organic molecules into very, very big macromolecules. So remember, there are 4 types of macromolecules: lipids, proteins, carbohydrates, and nucleic acids, or what we call polymers, right? Which are basically just repeats of very, very small organic molecules. How is that going to happen if there's nobody around to assemble them? Third, we need to go ahead and find some molecule that can assemble itself. I mean, like I said, there's nobody around there to put things together, so some molecule has to have the ability to self-replicate. Or to go back to our analogy, what came first, the chicken or the egg? We have to find a chicken that can make another chicken without there being an egg. And fourth, packaging of membranes with different internal environments. So this is really weird. For a cell to get going there, right, it has to then form a membrane, so what is inside is different than what's outside, right? But how could it form a membrane around itself if it didn't exist yet? That's a really weird idea. So you can't have it inside till you have a membrane, but how do you have a membrane without an inside? It's a very, very strange idea. Now what I'm going to do is I'm going to show 4 different ways that these 4 things could have happened. That doesn't mean that's the way it happened, but like a good scientist, I'm going to provide data, possibilities, so that there might be a reason to think this is what's going on. Okay, first of all, how could we make organic molecules from non-living inorganic matter? Well, Miller and Urey demonstrated this, or Urey and Miller, in their very famous experiment. And here's what they did. They went ahead and took a flask, and they filled it on up there with all sorts of goodies, right, that they thought would be inside a reducing environment. So what might be inside the reducing environment? Well, they filled it up with gases, like methane, hydrogen, and ammonia. So those guys go inside. And then they added very hot vapor water. Why is that? Because they thought the primitive Earth had a lot of steam. Okay, and then what they did is they ran an electrical spark through it. So now we go ahead and we take lightning in the form of electrical spark and we zap it. And then they went ahead and they waited about 2 weeks or so, right? And after 2 weeks, they noticed that something was going on in there. There was kind of a goo that was forming in the bottom of this flask. And they drained the goo out there and they analyzed it with a machine called an HPLC. And surprise, surprise, they found simple organic compounds. They found amino acids and they found sugars in there. Not only did they find them, but they were in pretty big concentrations. I think it was about 1 to 2% of it actually contained those organic molecules. Now that doesn't mean that this is the way it happened, But what they clearly demonstrated, here's the factual information, you can make organic molecules from inorganic molecules spontaneously just by zapping them with electricity. Okay, so that satisfies our first condition there. We got this one done. Now let's take a look at the next one. Now that we have organic molecules, how could you assemble them into long and long polymers? Okay? Well, this one's kind of tricky. You'd have to find a place on the primitive planet where such a thing would happen. We got to put energy in there to build bigger and bigger molecules. One place this might have happened is deep underwater. You find these really interesting fissures in the ocean floor, and they're attached to channels, and hot gases are bubbling on up there. These are called underwater hydrothermal vents. So methane and ammonia gases are pumping their way up through here, right? And then they mix with the colder water, and it goes back and forth like this. And it actually starts to form very interesting structures on there. They have noticed that this provides energy that puts molecules together. We found really interesting things down there. So one place this could have happened is underwater hydrothermal vents. Another place it could happen is volcanoes. Once again, in volcanoes, you have gases coming up, and they get in contact with lightning that could then create polymers. And there's been some people who have also said that it may have come from outer space. This is what they call the pandemic theory, that comets and so forth may have brought in polymers, large organic molecules. Okay, so now we provided two pieces of information for two of these, right? So assembly of molecules could have happened in hydrothermal vents, for example. Now let's look at the third one. Is there a molecule that is self-replicating? Let's take a look here. Well, there certainly is. It's a really common molecule. We know it, RNA. RNA is a very, very simple molecule, much more simple than DNA, since it's usually single-stranded, and it is what we call autocatalytic, which means it can start reactions on itself. And the way it does it is this. RNA can loop around on itself like this, and then it can cut itself. So it cuts itself right there, and therefore it can cut itself into two pieces. It does this on its own. So it becomes two smaller molecules. This is called autocatalytic, the ability to splice itself. But here's another thing. RNA can also be self-replicating, right? So if you have an RNA molecule, it can actually make a copy of itself, right? And then they separate off into two pieces of RNA. RNA can copy itself. And here's actually a third interesting function. RNA can be an enzyme. Now, typically, we always thought that enzymes were proteins, but RNA can be its own enzyme, and we call it a ribosome. Now, why is this really important? Here's the way we always thought things worked. DNA, which is in the nucleus of the cell, right, is then transcribed, made into RNA in the nucleolus, and then RNA leaves the nucleus and goes out to the ER, and then it is translated into proteins, right? That's the way we always thought things happen, right? There's only one problem with that, right? How did you get proteins to do all this if we never had RNA in between, right? So you had to go from here to here to here to get proteins, but you need proteins to do the first step here, to make DNA into RNA. So how did you do that? And there's a lot of biologists who are trying to think, you know, maybe the first molecule was RNA, which was able to autocatalytically make itself into a protein, because it was also an enzyme, and then maybe RNA, as time went on, became DNA. Maybe RNA was the first molecule because it could also act as an enzyme. Okay, let's go back here. Now we've established three pieces of information. We found a molecule that can make things on its own, and that is RNA. Let's go ahead now and look at the last one. How do we make membranes? Well, the first cells would have to have very, very primitive membranes, and here's how you do it. Remember we talked about phospholipids, how lipids have an ability, if you throw them into water, to kind of come together spontaneously to form a bilayer membrane. So there's properties of different molecules that let them do that. Lipids, if you throw them into water, automatically make a bilayer. So you would have, if you had fats, would automatically come together and make a very primitive cell like that, right? We call that primitive cell, sorry about this word, a coacervate, a coacervate. We also find interesting that proteins can do the same thing. If you have proteins and you throw them into water, right, like this, they will also come together because a lot of proteins are hydrophobic. They don't like water, just the way fats don't like water, and they will also form primitive cells, and those primitive cells are called microspheres. Together, this is the answer they usually ask for on the test, primitive cells, basically just membranes, right, are called protobionts. And once you've got a membrane situation, then all sorts of stuff can start going on here, which is separate from the outside. Okay, and now we've gone ahead and we've established a way that all four of these conditions could be met to start the first primitive cells, protobionts. Does that mean that it was actually this way that things happen? Not at all. But it could have happened this way. And so, if you look at the last one there, primitive cells, this is operance theory, right? Operance theory says we could have primitive cells which could spontaneously form from organic molecules. So we've met all four of them, right? Uri Miller showed we can make organic molecules from inorganic. We know that polymers can form from hydrothermal vents. Amino acids, therefore, could be put together in proteins. We found that RNA is a self-replicating molecule, which eliminates the need for proteins. And we found that we could have self-assembly of molecules because of their hydrophobic properties into primitive cells called protobionts. Okay, one last thing here before we end our discussion. There is another idea then of where we went after we had primitive cells. So a lot of people used to think that primitive cells, well, they just evolved over millions of years and they became more sophisticated. But there was a great biologist at Cornell University, and her name was Lynn Margolis. Here she is. She just died recently, unfortunately. And she came up with a different idea. And the different idea was focused on her work on mitochondria and chloroplasts. Now, here's what she noticed when she looked at mitochondria and chloroplasts. She noticed some very, very strange things. First thing she noticed was mitochondria and chloroplasts have their own DNA. That's very strange. We always thought DNA was only in the nucleus. Not so. We found DNA in mitochondria and chloroplasts. The second thing she found out was the DNA inside them is not a chromosome. It's circular naked, a lot like you'd find in a bacteria. She also found that mitochondria and chloroplasts have double membranes. That's really weird. Not one membrane. All organelles inside have one internal membrane. This guy has two. And finally, they have ribosomes. Well, there's lots of ribosomes inside. That's not anything unusual. But they're the bacterial type, the 70S, not the 80S like we'd find in there. Now, why would mitochondria and chloroplasts have all these sort of features that made them look like bacteria? And here's what she said. Because they once were bacteria. Her idea is that primitive cells engulfed bacteria. And instead of grinding them all up, they incorporated them. Now, remember we talked about how bacteria have, some of them have the function to do cellular respiration, and some of them have the function to do photosynthesis. Well, if you had a primitive cell, a protobiome, right, that would engulf a bacteria that could do cellular respiration, maybe that bacteria eventually became a mitochondria. That's where they came from. Or suppose it engulfed a bacteria that was able to do photosynthesis. Maybe it eventually became the organelle, a chloroplast. And that's why they look like bacteria. Now, this theory is a whole new way of looking at things. This is called the Endo symbiotic theory. And what she is saying is that the way cells became more and more sophisticated was not by slowly evolving, but to incorporating, to working together with other different bacteria, and they each brought in features and they made big leaps in a very quick time, and that's how they evolved. This is a totally different way of looking at it. So, we looked at 4 ways that we could have had primitive cells, protobiomes, and now Lynn Margolis is saying this is how those cells could have then come together endosymbiotically to become more and more sophisticated.